Another type of ELISA is known as sandwich ELISA. Sandwich ELISA typically requires a pair of antibodies to detect any particular antigen. This happens because of the epitopes, that the two antibodies, they are specific for different regions of the antigen. So in fact we need two different antibodies having two different overlapping parts or epitopes which can detect the same protein. In that pair, one antibody is known as capture antibody. The second one is known as detection antibody. Capture antibody is basically the one which is coating that particular polystyrene plate well. This capture antibody binds with the antigen, and then we have the detection antibody that will detect the antigen in the sample. Once we add the sample in the well that is coated with the capture antibody, that capture antibody will hold the specific antigen because of its specific segment in the antigen. And in the second step, we are going to add the detection antibody, which is also primary antibody, but this is complementary, or this will recognize a different part of the similar antigen. Then on the basis of method of detection, whether we are going to use the detection antibody that is coupled with an enzyme, this sandwich method can be direct sandwich ELISA, or if we are going to use an additional antibody, that is secondary antibody which is coupled with enzyme, that is going to be known as or referred as indirect sandwich ELISA. The sandwich ELISA differs from the conventional ELISA in the sense that it uses the two antibodies as compared to the other one in which we only use one primary antibody. This figure shows you the difference between direct sandwich ELISA and indirect sandwich ELISA. In direct sandwich ELISA, the primary antibody is coupled with the enzyme. In indirect sandwich ELISA, there is the capture antibody, there is the detection antibody, and then there is the secondary antibody that can detect the primary antibody or the detection antibody. This method of sandwich ELISA has further advantages, and for the reason that we are using two antibodies in this particular method, the specificity is quite high. This is highly specific ELISA, and we use this method for some complex samples to detect our required antigen. And for this purpose, for this method, the purification of the sample or the antigen is not required.